So with that, I will talk about something that is hopefully a positive thing for the future. And I'm asking the question whether there's any clinical utility in knowing about common genetic variation in your genome. What I'm not discussing, all of the work that goes into understanding insights into biology, courtesy of having large-scale genomic data linked up with phenotypes, all of the excitement about how this is going to or is changing drug development, all of the work that some people here are doing on applications of Mendelian randomization. I'll give you one exception. I'm not talking about rare variants in single genes, so that's the province of high-risk clinics and undiagnosed disease clinics, and I'm not talking about somatic mutations. So this is just a piece of genomics. I couldn't resist, since this is the home of nutritional epidemiology, just letting you know about something that I think is interesting in terms of Mendelian randomization support for higher dairy intake being associated with reduced risk of colorectal cancer. The pooling project of diet and cancer way back in 2004, In-Young Cho and others did this meta-analysis of dairy and colorectal cancer, a lot of heterogeneity, but a significant inverse association between dairy intake. And some of you may have seen this already because it's been published for over a year, but a meta-analysis of UK Biobank and FinGen has shown that the genetic prediction of dairy intake, mainly courtesy of the lactose intolerance phenotype, has almost exactly the same point estimate, showing that people who are able to eat more dairy, genetically proxied, have a lower risk of colorectal cancer. So perhaps some causal evidence. I'm only showing this because I'm aware of much larger scale data from the GECKO consortium, about 20 or 30,000 cases, that shows exactly the same thing. So I think we can sort of take that to the bank. Now, it's up to nutritional epidemiologists. Is that the dairy? Is that the calcium? And I know a lot of people are interested in the microbiome, and so I know you're already looking at some of the impact of dairy intake on microbiome and how that might affect risk of either colorectal adenomas or cancer. So I'm talking about common variation, and so the first thing is, well, what is common? Well, it depends on the population because there's heterogeneity across continental ancestries for many genetic variants. The usual definition is something that's 1% of alleles or 2% of people in a population. And it's important to distinguish between clinical utility, so basically is a medical test or practice actually useful, versus what we tend to study in epidemiology, which is clinical validity. Is a test actually predictive? And so most of what I'm going to show is clinical validity, and I'm going to ask the question how we get across the valley of death or make the bridge to utility. I'm getting a lot of feedback, but is that irritating people? Or okay, so we could turn this down or turn it off, and I'll speak into that. So can you hear me? Okay. I hope the people out there can hear us too.
No, I'm not sure the webinar is.
Okay.
I would keep it on, perhaps put it a bit lower.
So anybody know how to turn this down? You can turn it on and off.
Increase the distance.
Maybe that was the problem. Okay. How's that? Better? Great. So you've all seen these diagrams. Sequencing is getting cheaper and cheaper, and we'll soon be at the point where a whole genome sequence is the price of any of these common medical tests that we do at the drop of a hat, even getting your teeth cleaned. And we'll be at the point where if you need to know any specific germline variant in your genome, rather than actually going after that specific variant, with one of the tests that Immaculata or Paddy's lab do, it'll just do the whole genome. It'll just be quicker. Which will raise the question, what are you going to do with the other 99.99% of the genome? We could throw it away, and actually it'll be cheap enough that you can just sort of do it again when you need it, or you can put it in a database and be able to access it on demand. It's worth noting that there's a large project in the UK now, 100K variants in infants, and that's basically to see whether the whole genome adds information to the standard mass spec screening for some of the rare inborn errors of metabolism. But they're extending that beyond that sort of classical area that, you know, anybody who's had a kid in the last 30 years, the kid's had a heel prick and that's been done, to rare variants that cause potential genetic diagnosis that might actually be an inborn error of metabolism and may or may not have something that is clinically useful, but the hypothesis is, well, this will potentially be generalizable and stop the diagnostic odyssey where parents have to go around the country looking for a place that will actually make a diagnosis of their child who has some problem that hasn't been diagnosed. So with those 100K infants, obviously you'll have the whole genome, and the question is that a database in which you'll look for other things that aren't necessarily diagnostic in infancy? The project consent is to just look at the diagnostic in infancy stuff, but it's just an indication of how fast all this is moving. And we've learned over the last 20, 25 years that you can essentially divide genetic variation up into the very rare stuff, so think of BRCA1, BRCA2 at early age, relative risks of 20, 25, and 30. Some stuff that's still rare, so you have to sequence to find it, but the effects aren't so large. The polygenic variants, so these have very, very small effects, but they're very common in the population, above 1 or 2%. In many cases, 60 or 70% of people carry a variant that may be associated with a relative risk of now down to literally 1.02. But collectively, these add up to polygenic variation. And then a few large effect variants. The classical example is an HLA variant that is associated with ankylosing spondylitis. But again, the variant is relatively common, but ankylosing spondylitis isn't that common, so the penetrance is reduced. So I'm going to be talking very briefly about this top, and then mainly about the polygenic variants. And so, again, everybody is aware that courtesy of the whole genome project, the HapMap project, making the database available, and then the genome chips, where we could reliably determine 100,000, 300,000, 500,000, now a million variants on a chip. We could basically take genetic epidemiology. When George Hutchinson was teaching me in 1985 and 6, it was all family-based, pedigree-based, SIBShips, etc. And we could do regular epidemiology, case control studies, nested case control studies, even in something like UKB, true prospective studies. Manhattan plot, looking for the tiny p-values that are associated with relatively small effects. First of all, with small sample sizes, 2,000 cases, finding one or two of these and thinking we'd discovered something completely novel and actionable. Over time, the relative list dropped very fast as sample sizes increased, and it was possible to access, with robust replication, variants that, again, literally have relative risks of 1.02 for heterozygotes, 1.04 for homozygotes. But you add them all up, and the genome is saturated with these. So you take the variants of small effect, put them into a model, and you come up with a polygenic risk score. People who have a higher handful of the risk variants. People have a lower handful or higher handful of the protective variants, and that gives you a polygenic risk score. Jamie?
Previous what was? I didn't understand.
Sorry, this one?
No, the previous.
I'm sorry, I'm sort of moving fast. So this is odds ratio on the y-axis and year on the x-axis.
Not the odds ratio of the same thing.
No, odds ratios of new findings as the years went by. So basically, when the sample sizes were small in 2007, we could pick up a relative risk odds ratio of about 1.3, and it's declined over time. Sorry, I should have explained that. And this is the 23 chromosomes painted with all of the bona fide genome-wide association variants for a large number there of different phenotypes, but now there are GWAS for 200 diseases or interesting phenotypes and conditions. So going back to the common stuff, maybe you'll be aware that the American College of Medical Genetics has a 73 gene list. So if you did a whole genome sequence, the question is, would you like to know of any variation in these 73 genes that they decided are robustly associated with particular diseases and are worth knowing if you choose to know them? So, for instance, the All of Us project in the US, if any of you, must be someone here in the All of Us project. Okay. So basically, all of us will do your genome, and if you wish, after some counseling, we'll tell you if you have a pathogenic variant in any of these 73 genes. Now, it's worth pointing out that if you're an adult having a pathogenic variant in a gene that predisposes to infant or very early onset disease, probably means that it's not that relevant to you. But, you know, even as an adult, it could be a BRCA1 or a BRCA2 that you don't know about. And so again, this is the way the field is going. All of us is a cohort study. All of us is returning results to people who want to know them. The question here is, are any of these common, or are we only talking about rare stuff? And the answer is, in the whole 73 gene list, I've only been able to find one bona fide pathogenic variant. And that's a relatively common variant that essentially underlies hereditary hemochromatosis. Nasty disease, if you're a drinking male who's at risk of hemochromatosis, because your liver gets packed with iron and the alcohol on top of the liver might give you cirrhosis. There's, in the UK Biobank, a 17-fold increased risk of liver cancer in men, not just men drinkers, but men who have a homozygote for the key variant. Women get a bit of a break because premenstrually, menstruating, running anemic, and so it's only when they become postmenopausal that they start to put the iron into their liver, pancreas, and joints. But the work in the UK Biobank, the person to look here for is David Meltzer, MEL-ZER, has shown that there is actually an appreciable high burden of disease in people who are homozygotes. But homozygosity, the variant, is about 5 or 10%. Homozygosity is about 1 in 160 of European ancestry people. So it barely meets my definition of common, because you really have to be homozygote for this to be a problem. HLA is too complex a region to genotype or sequence to be in the ACMG list. There's a few special populations that you'll be aware of, particularly the Ashkenazi Jewish population with a couple of founder mutations that are getting towards collectively 1% of the population that are breast cancer genes. And then there's a special case of ApoE4. And people woke up 25, 30 years ago probably, mainly cardiovascular researchers who were looking at ApoE and had the genotype and suddenly discovered that it had been established 20% of European ancestry people who carry one copy of the ApoE4 allele were at 2 or 3-fold increased risk of dementia, Alzheimer's disease, and homozygotes, let's say 5 to 6. It's one of Bert's papers, I remember, was one of the first prospective studies, I think it was in the BMJ, to look at cognitive decline. All of the advice here is you don't want to get your ApoE4 status because there's nothing you can do about it. There's plenty you can do about it. So this is some data. This is Iris Kim working in the Nurses' Health Study and looking at what the Nurses' Health Study has reliably documented, which is cognitive decline as opposed to Alzheimer's per se, and asking if I'm an E4 carrier and I am hypertensive, is my cognitive decline the same or faster than if I'm an E4 carrier and I'm not hypertensive? And so this is an observational study. So if you've got hypertension and you don't carry the allele, no significant reduction in your cognitive function over time. If you're an E4 carrier, though, substantial, steeper decline, maybe a little steeper if you're hypertensive. Again, observational design. But if you're actually a treated hypertensive, drug plus, you have a slower rate of decline in this study than if you're untreated hypertensive. So if you were E4 positive and you're hypertensive, you should probably try and reduce that blood pressure. Better studies have been more comprehensive. So this is a recent study, cohort study, and looking at 17,000 people followed over time and using this score, which amalgamates a variety of mainly cardiovascular risk factors and scores you as favorable with respect to those risk factors, intermediate or unfavorable. And then if we just look at this at the top here, these are the E4 carriers in the bottom 3 rows. And if you have a favorable environmental and lifestyle profile, your relative risk of Alzheimer's is elevated, but it's less elevated than if it's unfavorable. And it's exactly the same for the other categories of the genotype. So this is pretty much what we've established, with some exceptions, and I'll talk about the drug idiosyncrasies. Genetic, environmental, lifestyle risk factors are independent, and basically the risks multiply. And the absolute risks then are going to be larger for healthy lifestyle if you're at higher baseline risk, courtesy of your genetic higher risk. But it immediately gets you into the sort of public health arena. Do you want to know your E4 allele? Because there are things you can do about it to reduce your risk or at least delay the onset of Alzheimer's. But, is that basically something that it's worth knowing and it's worth asking people if they want? Or do we just do the same thing we've always done, which is basically population-wide messaging that high blood pressure, obesity, all of those lifestyle risk factors are not a good thing, and so it doesn't really matter if you're at elevated risk at birth. We should just have a one-size-fits-all broad brush message. So common variants, there's not much in the genome that's established that is clinically useful. And that question there at the bottom is not a matter of clinical utility, it's more a matter of public health policy. Just very briefly, the usual argument against knowing your ApoE4, Deborah knows as well, is, well, it'll just create a lot of anxiety. To the extent it's been studied, albeit small studies, Robert Green and others, if people are counseled appropriately once they know their ApoE4 status, people who are positive may experience a brief increase in anxiety, but 6 months later, that's kind of washed out. So done right, nobody is lying awake at night after a few months worrying about this, and it does potentially alert people to a need for modifiable lifestyle choices if they aren't already in the healthy department. The killer app for the genome was supposed to be pharmacogenetics. The idea here is that we just give drugs to everybody empirically. We titrate those drugs according to effect when we've got the option of doing that. So we titrate, does it work for blood pressure? If it doesn't, we'll move you on to another drug. If that doesn't work well enough, we'll add a second drug or a third drug. The vision of pharmacogenetics, because we have established that there are a lot of genetic variants that influence the metabolism, absorption, excretion of drugs. Could we get you on the right drug in the first place if we could predict from your genome what drug you should start with, what dose you should start with? Would this eliminate some of the trial and error in prescribing drugs? And the pharmacogenetics people talk a good game. About 6% of people in hospital are there because they've got a drug interaction or a overdose or a toxic reaction that we could have avoided. And this is a diagram, the paper's from 2013, but it goes right back. This was a market that was going to explode. That would be a rationale for doing large-scale genotyping because there's all these genes, I'm not going to sort of describe what they are, that work across a wide variety of different diseases. There's even a randomized trial in Europe now that basically divided people who were about to be prescribed a drug into a genetically informed choice of drug and dose and people who were usual care. So usual care is blue, event rate are the adverse reactions, and a significant reduction in adverse reactions if we did pharmacogenetic-based prescribing. Those of you who will know this, Alec and others, some of these genetic variants aren't easy to genotype. So currently you would be charged $300, $400, $500 to get a pharmacogenetic panel. Once we have the whole genome for $100 or $200, it's probably going to be accurate enough to make the same diagnosis at lower cost. Again, it could be in your medical record. So if I show up at the Brigham emergency room in a coma, it's already there. If somebody wants to prescribe me a drug that could be influenced, and a very high proportion of commonly used drugs have an established efficacy variation or adverse effect variation based on their metabolism courtesy of the genetic prediction. It even, as of last week, affects whether I should be taking, if I have mild cognitive function, the new generation of drugs that are now approved for people with mild cognitive function to try and delay advanced Alzheimer's. And this raises the ethical question. So it turns out that the ApoE4 carriers, we talked about them earlier, if you give them these anti-amyloid drugs, they're much more likely to get a brain bleed. Brain bleeds can be devastating. And so should everybody have been told their genetic status at the start of these studies, because that was an established association. And the company that did the studies without telling people is criticized in this article for essentially not having weeded out the people who they probably wanted to study because they were at the higher risk of Alzheimer's. So it does get kind of complicated, but I would argue that, God forbid, anybody here needs any of these drugs soon, you probably do want to know your ApoE4 status. And so again, this was the paradigm. Current reactive medical model, try a drug, try a different drug, try a third drug. By that time, actually, the problem may have gone away of its own, or move to a predisposition screening and use the right drug at the right time. So why hasn't this caught on? Well, Who pays? There is issue with standardization of the genotyping. Much of the information is based on people of European ancestry, and so these genes are among the genes that vary substantially across continental ancestries, so we need more information from people of African and Asian ancestry. I think the big problem is that actually it's pretty cheap to prescribe a drug that's a generic drug, and if that doesn't work in 3 months, up the dose, change the drug. That works for things like hypertension and other issues, other phenotypes, other problems you're trying to solve that aren't urgent. It doesn't really work for cancer chemotherapy because you want to get on with that and you don't want to decide after 3 months, well, that's not working, let's use a more effective drug. Patients and their medical advisors have a hard time with complex probabilities versus just sort of dichotomous decisions. And the other thing is there's a distinct lack of decision support materials. So if you're the prescriber at the moment, you're prescribing, we now have good decision support materials that say, well, no, you shouldn't prescribe that drug at that dose because your patient is on this other drug and that'll interact. But nothing I've seen really effectively does that for the genome. So this is really a problem. The bemused pharmacist, and here's my genome on a chip. What do I do? So this brings us to polygenic scores, and basically you will all understand how that works. Now we have dozens, hundreds, even thousands of variants that have these small variations in risk of a particular disease. We can put these together in a continuous risk score distribution, and like most biomarkers, we sort of have a normal distribution of risk that we find in the cases that might be higher than we find in the controls. And they actually sort of translate into fairly robust and interesting differences in lifetime risk if you use a study like the UK Biobank and follow people over time. For a disease like type 2 diabetes, the first issue is that we can actually do this very well based on non-genetic risk factors, mainly body mass index, waist circumference, a little bit of family history. So this is a score that's used in the UK, the so-called Leicester score, just a clinical algorithm. And here are people who are high, moderate, a bit increased, and low on the basis of the score. It's really driven by BMI and waist circumference, and their cumulative incidence over time in the UK Biobank. So like all new risk factors, if you've got a really pretty robust set of predictors, it's very hard for your new predictor to add much to the predictive value. I just want to make one sort of tangent here, and this is a slide that I should get remade because it's a little bit more complicated than it has to be. But those of you who use UK Biobank, you're probably used to using the disease incidence data out of the 500,000 people in UK Biobank. And that's great for things that get you into hospital because the disease information is by and large based on the HES data, the hospital episode statistics data. So if you have a heart attack or a stroke or you fracture your femur, you're going to wind up in that HES data. However, if you're diagnosed in the community with something that doesn't necessarily involve immediately a hospitalization, Parkinson's disease, type 2 diabetes, or dementia, you don't show up in the UKB disease file until you get admitted with a stroke, MI, or fractured femur. And then they code your Parkinson's, type 2 diabetes, or dementia as a secondary diagnosis. In UKB, they were only able to get the primary care data for 45% of the population up through 2016. People can't hear? We're on. Oh, the light's gone off. Maybe the battery's gone? Yeah. talking, and if we can, yep. Sorry, battery's gone. We looked at this in the UK Biobank, and in blue here, we have the hospital episodes only available to everybody. In the red, we've got the cumulative incidence of these diseases. If you include the GP data, that's available for about half the follow-up for half the people. And as you'd expect, there's a much higher cumulative incidence when you can include the GP data where the ambulatory diagnoses are first made and recorded. This is important to know because I've seen a lot of UKB papers that make the assumption that you can look at these ambulatory diagnoses and rely on the hospital episode statistics to give you cumulative incidence, particularly if you're looking at a polygenic risk score and you're trying to calculate risk over time. This was published in European Journal of Epidemiology, and I had to thank Bert for accepting it because I'm told it has made a difference. And this is a quote from the new Minister of Health in the UK, Wes Streeting, who basically says, that's why I'm directing NHS England to take responsibility for where people have given consent, making the primary care data available to consented cohorts. So I'm told that this was somewhat influential, and that's going to mean that studies like UK Biobank and other UK studies are going to have much, much more complete information on these diseases that don't necessarily cause a hospitalization. The other issue I referred to earlier, we're very aware that we're on thinner ice when we're talking about populations that aren't European ancestry, because 85% of the worldwide data that's in the studies that's used to give rise to the polygenic risk scores have been European ancestry, done in Europe, done in North America, done in Australia. But it doesn't necessarily mean that the information doesn't work in other ancestries. So here's an example for South Asians, type 2 diabetes, red, high PRS, blue, low PRS, teal, median, and courtesy of the fact that South Asians have a higher risk of type 2 diabetes, you actually get a bigger discrimination between top and bottom. So each of these diseases you have to look at separately. And it's not true to say that the European-derived polygenic risk score doesn't convey any information about other continental ancestries. For many diseases, it does. For many diseases, it doesn't. And it doesn't tend to do well with African ancestry. Talking very briefly about breast cancer, I'm going to move through that. And just quickly point out that this doesn't stop at the epidemiology of first diagnosis. So here is a study looking at whether people who were on a blood pressure medication on the left, on a statin on the right, had their blood pressure in one case or their LDL cholesterol controlled. All of these people are on treatment. And people in the highest polygenic risk score had the highest relative risk of not being adequately controlled for both blood pressure and LDL cholesterol. So you can make an argument that, again, this is sort of pharmacogenetics. The polygenic risk score identifies people who are going to need a higher dose or a different drug. Now, there's been a lot of hate directed at polygenic risk scores, because if you look at it through the risk of a screening study, how well does this discriminate people who become cases and or have the disease and don't have the disease, you don't do that well. But this is pretty much, I learned this in Walt Willard's class. Here we have serum cholesterol. Here's the original Framingham data, the distribution of serum cholesterol for people who went on to have heart disease, and in blue, the people who didn't. You don't get great discrimination, but it's still a legitimate risk factor that you can add to your armamentarium. So polygenic risk scores are risk factors. They're not diagnostics. If you analyze them as diagnostics, they fail. I haven't gone over all the data, but they often do better than the previous environmental or clinical risk score. They always add. In some diseases, they add a little. In some diseases, they add a lot. we still need more information from non-European ancestry. And there's an argument that at younger age, when you don't know when I'm 20, you don't know whether when I'm 40 and you run the Framingham score, I'll still be a smoker, whether I'll be obese, whether I'll be hypertensive, that the polygenic risk score could tell us a lot more information at early age. So why hasn't chip genotyping become more mainstream? Well, there's a whole list. Partly, we're waiting for the whole genome scans because chips haven't become a sort of marketable product. It's still a research product. There's very few actionable single variants. We're still not able to incorporate the pharmacogenetic work because we don't have the decision support tools and the knowledge to do that. With any new technology, it takes time to get into the clinical environment. And then sort of ethically and psychologically, if I want to know about one risk and there's a good clinical indication for that, do I really want to open up Pandora's box and find out about several dozen other risks that I may or may not be able to do something about? So there's two valleys of death here to take this information into the clinic. The first we're actually pretty good at, the clinical validity, doing the genetic epidemiology through the large databases and consortia, establishing the associations. Where we're still very, very early days is the next step, which is taking that information and asking, is it useful in clinical practice and health decision making? So I very quickly want to introduce you to a new study. People still ask, well, why did you give up that tenured position at Harvard to go to UK where there's a mandatory retirement age that I'm rapidly approaching? And the answer was this was the sort of gleam in the eye, and we've sort of got it up and running. Our Future Health, the aim is to get 5 million volunteers and try and make that as generalizable across the population as we can. Unlike something like UK Biobank, we're not just building a resource for research. We intend to offer people personal disease risk if they're interested, and then recontact people who might be interested in doing additional risk-stratified studies.
I'm not sure, but you can try it again. Maybe try speaking into it.
Nope, sounds like it's working. Great.
I think it's, I would just, it's like inside the shoe.
Inside the shirt.
No, sorry. Like, how to do this way.
Got it. Thank you. Yes. So this is all very much a work in progress, and it's all been made possible through about 45% government funding, 50% industry funding, about 5% charity funding. We only started 2 years ago. We got delayed during COVID, and basically the secret to success here is it's a very short enrollment visit, but it's essentially the same as the All of Us cohort in the US. Baseline questionnaire online, consent online, 2 tubes of blood, and some anthropometry, blood pressure, and a point-of-care lipid value. The secret to success here was going into community pharmacies and really making enrollment possible within a couple of miles of where the majority of people live. And so as of earlier this month, we hit 1 million full participants that have given blood and had a clinic visit. We've got almost 2 million people who've volunteered online, done the consent. Notionally, they're waiting for an opportunity to do the research visit near them. So looking at this, you could say we've got 4 million to go or 3 million to go. It's happened super fast, and that's because of the resources available in the UK and this very, very widely distributed enrollment scheme, mainly online, quick visit at a place near your residence. I won't go over the genome chip, but basically standard Illumina chip supplemented with variants from the various disease-specific consortia. We're getting information from the HES data, which I explained earlier. We're hoping to get the primary care data courtesy of the Ministry of Health and the paper in European Journal of Epidemiology, all of the cancer treatment and other data. This is sort of a key slide based on the first 1.5 million consented people. Like UK Biobank, we get more volunteers who are female. Unlike UK Biobank and unlike most of the biobanks, we are enrolling from age 18 up. So we've got a substantial number of younger people, so the mental health people are pretty excited about that. We've got a decent and better than previous studies enrollment of South Asians, and we're running about 50% of the population prevalence of people of African ancestry. And like many studies, we do better on people who are higher SES, fifth quintile of the deprivation index versus low, but we're still doing better than many prior studies. The good news is you can get all this data, so that's the point of this part of the talk. Just like UKB, application access, right now you can get almost 1 million questionnaires with linked hospital episode statistics data, and 330,000 have a genome array available. By December, that should be about 650,000 people with a genome array available. So technically it'll be bigger than UKB. Of course, it's pretty much all prevalent disease, very little incident disease at this point. Just like UKB in a lot of projects now, instead of dishing the data out to anybody who asked for it, you now have to come to our trusted research environment. That involves a certain amount of training, that involves only being able to run programs and export summary data and tables rather than the raw individual data, but this is the way the sort of whole field has moved. And this is just a summary again of what we're hoping to do in the future. Recontact participants to invite them to receive some of the genetic information. I'll be interested to see what the uptake of that is, and to be involved in additional risk stratified deep phenotyping or randomized trials. With that, I'll just thank my colleagues at Our Future Health, and I skipped over some of their contributions, but the guys in the group in Oxford. Thank you.